And with the rest of the time, we start another lecture of Enterobacteriaceae. That is a long word. You can see that Enterobacteriaceae. It's not easy to pronounce. Let me show you how to pronounce this long word.
Enterobacteriaceae. Enterobacteriaceae.
Can you hear it? I didn't hear it. Okay, let me play it again.
Enterobacteriaceae. Enterobacteriaceae.
Okay, so Enterobacteriaceae are a big family. It's not a genus. It's not a species. It's a big family, including a lot of genera. And in this word, entero means enteric, so that is related to our intestinal tract. And these bacteria are gram-negative, and they are bacilli. They are rod-shaped. So these are the big family of enteric, gram-negative, rod-shaped bacteria. Okay. A quick summary of this big family. We can check their common properties. First, all of them are gram-negative rods or gram-negative bacilli, and their natural habitat is the intestinal tract of human. So they are living inside our body, inside our intestinal tract. This family includes many genera, including like E. coli, that is the Escherichia. I cannot pronounce this word; it's long. Shigella, Salmonella, Enterobacter, Klebsiella, and a lot. So it's a big family, including a lot of genera of bacteria, and some enteric organisms are part of the normal flora. We learned the normal flora, and in our digestive tract, in the intestinal tract, there are also a special group of bacteria. They are residing there, so that is the normal microbiota. Some of those bacteria are part of the normal microbiota, and they are living peacefully with us. They don't cause disease. But once those bacteria translocate to other place of our body, so if they move to other tissues, they might cause opportunistic infection, such as E. coli. They don't cause disease in the digestive tract, but they can cause infection in like genital tract. So that's different. Some are regularly pathogenic for humans, so means that in this big family there are some professional pathogens. They can cause disease in humans, such as Salmonella, Shigella, which can cause disease in humans, and even E. coli. When we say E. coli, that's a species name, and there are also different types of E. coli. You can say they are different strains or different subtypes, subspecies. While some species, subspecies can cause infection in human, and later we'll introduce those pathogenic E. coli. For morphology, those big family, all of them are short gram-negative rods. So which means that you cannot differentiate those bacteria only by gram staining, because they look similar. They don't form spores, so no spores. motile. Some of them, or a majority of them, are motile via flagella, but Shigella and Klebsiella doesn't have flagella. They are not motile. And for culture, you can culture those bacteria on a lot of media, common media. And for example, on the LB plate, they can grow. They can form circular colonies. And the colonies, usually they are smooth with distinct edges. They are aerobic, which means that they prefer the presence of oxygen to grow. But some of them are also facultative anaerobic, which means that without oxygen they can also grow. That is not surprising because in the intestinal tract, that's the low that's a condition, that's an environment sorry, in the intestinal tract, that's the environment with very low level of oxygen. So that makes the bacteria evolved the ability to live in that environment. So without oxygen, some of them can also grow. They don't produce oxidase, so that's oxidase negative. For growth characteristics, we can add in different substrates in the when we culture the bacteria, and some bacteria can utilize the substrate or can break them down to make some specific molecules. And when we started this, we started a growth characteristic. From this, we can derive useful information to determine the type of bacteria. For example, the carbohydrate fermentation test. Different bacteria can utilize different carbohydrate, so that's sugars, different types of sugars. For example, E. coli, it can utilize both glucose and lactose. It can ferment both glucose and lactose and produce acid and gas. If it can produce acid and you add in pH indicator into the reaction, and you will see the color change. And if there is gas production and you put the bacterial culture in this big tube and you put another small tube inside the big tube, and the small tube can collect gas bubble in it, and you will see that this bacteria produced gas. So, and this is E. coli. It can produce both acid and gas. But for Shigella and Salmonella, they can only ferment glucose but not lactose. And when they ferment glucose, there is only acid produced but no gas produced. And based on this, you can get some information about the bacteria. Their antigenic structure, we know that they are gram-negative bacteria, and some of them have capsules and they have flagellas. So there are some important antigens. For example, the O antigen, that's the lipopolysaccharide. It contains the O side chains. So this is one we learned a lot previously. The polysaccharides are the components of the outer membrane, and it's resistant to heat and alcohol. You cannot denature these molecules. And the antibodies against O antigen is IgM. So it stimulates the immune system to produce IgM. While another antigen is K antigen. That's the antigen on the capsule. So capsule antigen, K antigen. It exists on some, but not all the Enterobacteriaceae. Some are polysaccharides, while some are proteins associated with virulence. Okay. A third group of antigen is the H antigens. So that's the antigen on flagella. It's protein antigen. You can denature or inactivate the flagella antigen by heat or alcohol. So it's a heat labile. And the antibody against the H antigen is IgG. So that's different. It's different from this one. O antigen, IgM, and H antigen is IgG. Okay. Okay, I guess we can have a break for about 10 minutes, and we'll be back in 10 minutes. And when we are back, we'll talk about the most important Enterobacteriaceae members, including E. coli, Shigella, and Salmonella, one by one. Okay, we'll be back in 10 minutes. Just now we introduced the general common features of Enterobacteriaceae, their morphology, and their growth characteristics and how to culture them. And now we introduce some of the important medical pathogens within this big family: E. coli, Shigella, and Salmonella. First thing, E. coli. Well, for a lot of strains or subspecies of E. coli, they are not professional pathogens. They are a member of the normal flora. But some E. coli, they somehow get the virulence gene from either other bacteria or from the environment, or they evolved by themselves. And those E. coli are pathogenic in the intestinal tract. So here we'll learn those E. coli pathogens. For general introduction, a lot of things we talk about, they are motile, means that they have flagella. They can ferment both glucose and lactose to produce acid and gas. They have O, H, and K antigens. They are a member of the normal intestinal flora. Some of them are pathogenic. And those normal flora, when they are in the intestinal tract, they don't cause disease. But once their location is changed, for example, they can infect urinary tract, or they can cause sepsis, they can cause meningitis. So those are the opportunistic infection. Another type of infection is pathogenic E. coli. They are professional pathogens, and there are different types of pathogenic E. coli. Here, their short name are included like E, PEC, ETEC, EI, EA, EH. And we'll introduce each type of pathogenic E. coli in detail. In these names, ETEC is short for enterotoxigenic E. coli, and the first E means entero, and T means toxigenic, and the last two letters EC means E. coli. So ETEC, the property of this bacteria is the T letter, the second letter, so it's toxigenic. And we know that toxigenic is related to toxins. So these bacteria can cause disease through toxins. They can produce toxins. This is a common cause of traveler's diarrhea, and also diarrhea in infants in developing countries. In a lot of the developing countries, such as in China, in India, and in like other developing countries, the hygiene condition or control is not strict, and so there might be pathogens in some food or drink which can cause infection, and ETEC is one of them. They can, once they infect human, they adhere to the epithelial cells of the small bowel. This is the epithelial cell, the model of the intestinal epithelial cell. This bacteria doesn't invade into the cell, but they stick to the outside of the cell, adhere to the cell. And there, they can produce toxins. There are two types of toxins. One is heat labile enterotoxin, heat labile exotoxin, short for LT, labile toxin. Another one is heat stable exotoxin, ST, and they can cause different change in the epithelial cell. Here's the mechanism of labile toxin and stable toxin. This is the bacteria adhere to the surface of the epithelial cell, and the stable toxin and the labile toxin are produced by the bacteria and can inject the toxin into the cell. And in the cell, they function differently. The ST can increase the cGMP, that's the cyclic GMP, while LT can increase the cAMP, the cyclic AMP. And those are the signal transduction molecules, or signal transduction mediators. And those molecules can, once the levels are higher, they can promote the epithelial cells to secrete water and chloride into the lumen. And that means that in the lumen there will be a lot of water because there is a high hypersecretion of water and chloride, and also inhibit the absorption of sodium to the cell. And if there are too much water in the lumen, it will cause diarrhea. So this is the reason why the toxin, ETEC toxin, can lead to diarrhea. Okay. A second one is EPEC, and in this word, P means pathogenic. So it's another group of E. coli. It's an important cause of diarrhea in infants. They express bundle-forming pili on their surface, promote the tight adherence. They adhere to the epithelial cell, lead to the loss of microvilli. You know, we have the microvilli like this on the epithelial cells. And once the bacteria adhere to the cell, they can damage the microvilli. And the result of EPEC infection in infants is severe watery diarrhea, vomiting, and fever, and cause severe disease. Okay, so this is EPEC. A third one is EIEC. In this word, I means invasive. And you know, invasive means that the bacteria can invade. And previous two bacteria, they are sticking to the surface of the cell. They are not in the cell. But for EIEC, they invade into the cell. So that is why we call it invasive. So the bacteria get into the epithelial cell. It produces a disease very similar to Shigella, so the disease caused by Shigella. And the disease occurs most commonly in children. Okay. Bacteria can invade intestinal mucosal epithelial cells. Okay, another one is HEC, and here H means hemorrhagic. Hemorrhagic means it's related to blood. Hem, H-E-M, is related to blood. It causes hemorrhagic colitis in the colon, hemolytic uremic syndrome. They produce a sugar-like toxin similar to the toxin produced by Shigella. The serotype of this HEC is O157H7. Just remember these numbers because this O157H7 is a very important and famous group of oxygens, and they are very important and dangerous E. coli pathogens. And in recent years, there are some outbreaks of E. coli, and a majority of them are O157H7, including the one happened in Germany about 10 years ago. This bacteria can produce a Shiga-like toxin, and this Shiga-like toxin can inhibit the protein synthesis and can also spread to the bloodstream and kidney, causing hemolytic uremic syndrome, EHEC. Okay, the last one is EAEC. Remember, the A stands for aggregative. Aggregative means the bacteria cluster together, aggregate together. They can cause acute and chronic diarrhea. They are characterized by their specific patterns of adherence to human cells. These are the human cells. They adhere to the human cell in the lumen. They can produce ST-like toxin, the stable toxin-like toxin, enterotoxin, and hemolysin, a lot of toxins, and cause chronic diarrhea. And you can see the special aggregation of the bacteria. It like breaks in the lumen adhered to the human cells. This is EAEC. Okay, so that's the different pathogenic E. coli. For laboratory tests to diagnose E. coli, we can use this procedure. You collect specimen from either urine, blood, pus, spinal fluid, sputum, or other material, and you make smear, and those smear you can check their morphology, and you can culture with either blood agar or other differential agar, and from those culture you can derive useful information. For treatment, well, E. coli infection does not have any specific therapy, but some antibiotics can be used, such as ampicillin, cephalosporin, and other fluoroquinolones. And those are the antibiotics you can try. But before you try the antibiotic, you should do the antibiotic sensitivity test or susceptible test. In recent years, there are some outbreaks of E. coli infection, especially in Western countries, such as in the one in Germany happened in 2011. It was in high school. A lot of students got infected and some died. Another one happened in USA. Actually, it happens a lot in USA. The recent one is from 2018. Here is a news video talking about that E. coli infection, and we can watch this video a little bit.
Big news, throw it all away. That is the message from the Centers for Disease Control and Prevention. If you have any romaine lettuce in your home or business, the CDC is linking that lettuce to a multi-state E. coli outbreak. Joining us trying to talk more about that is Dr. Mikhail Varshevsky, a.k.a. Dr. Mike. And Dr. Mike, it's great to see you.
Great to see you.
Talking about this, and it's worrisome.
Yeah, it's an important story.
The CDC say get rid of all romaine lettuce.
Yeah, especially Thanksgiving is upon us. We're going to be gathering together soon with the families. We're going to be eating. Get rid of all of the romaine lettuce, whether it's chopped, whole heads, it doesn't matter. Get rid of it because we don't know the exact source yet. The way we go about this process is we find people who have E. coli outbreaks in multiple states. Then we ask them questions. What have you been eating? And so far, that common denominator has been romaine lettuce. The E. coli, the type of bacteria that's getting people sick, is genetically similar to the E. coli that was found in the outbreak in 2017 of romaine lettuce that was mostly made in Canada. Now, why is this scary to us doctors? Because E. coli can then become an infection, which then leads to kidney failure, and especially in those who are younger, older, and those who have a compromised immune system. So it's very scary to us doctors.
How does this happen? I mean, how did the E. coli get in the lettuce?
So E. coli, it's not pleasant to say, but it lives in the intestines of cattle. And because cattle live near romaine lettuce producing plants, they're found in the fertilizer. This E. coli can then lead to being on the lettuce. But then most people say, well, I'll just wash the lettuce. It's not a problem. The CDC has done studies on this.
Can't wash
they wash lettuce. The bacteria still stays on.
Not in term, and particularly with somebody who has a compromised immune system, washing it does not get rid of the bacteria.
There are other vegetables like that. I mean, there's a list of the dirty dozen that no matter how much you wash them, like potatoes, tomatoes, they'll never, whatever's on them, they'll never come off.
That's why I stay away from all vegetables. As a policy, I just stay away from vegetables. It's a lot safe. Short-term safe, long-term meat you cook and you're fine.
Well, this Thanksgiving, I'm giving you a pass. There's no donut recall, so go nuts with this food.
Well, we have two ideas to talk to you about. What the CDC first says that more than 3.6 million middle and high school students used e-cigarettes in the past 30 days. Juul vaping devices supposedly designed to...
Okay, so the first part of this news is on the E. coli outbreak in USA. Someone said you know him. You mean that expert talking about E. coli? Yeah. So, yeah, E. coli. Oh, okay, great. I think that guy is working for CDC in USA, and he's maybe an expert there. Okay, so E. coli infection happens a lot in America. I think for one reason is because of the lifestyle there, because people prefer to eat raw lettuce, raw salads like that. In almost all salads, it contains lettuce, and lettuce is harvested directly from soil, from the ground. And you know, in the soil, in the field, there are a lot of bacteria, including pathogens. Especially in recent years, there is a trend of or popularity of organic food. For organic food, organic vegetables, they are not using chemical fertilizer, but they are using like organic fertilizer. Usually it's waste or feces from either human or cattle. And in those wastes, there are a lot of bacteria and pathogens, and that increases the opportunity to contaminate lettuce there. And in the news, you also heard that even though you wash the lettuce, it doesn't help. It cannot remove the bacteria sticking on them. So that is why people might get infected there. But the good thing is that there is a very good system to monitor the market. Once there is product with problem, the product will be recalled very quickly all over the country. Um, so that's the news. That's the news from Fox News. Even though I don't like Fox News, based on their behaviors in recent several years, and also their action on the election, the recent election. Yeah, so that's the Fox News. Okay. The E. coli treatment. The next one we will talk about is Shigella. Shigella is also a gram-negative bacteria. It's a slender or very thin rod, as you can see from this image. They are also gram-negative rods and slender. They are facultative anaerobic, meaning that they prefer oxygen, but without oxygen they can also grow. They grow best aerobically. They are not motile because they don't produce flagella. It can ferment glucose but cannot ferment lactose. It produces acid but not gas. Most strains do not ferment lactose, except for the Shigella sani. I think this is a very special species of Shigella which can also ferment lactose. These are the general features. There are different types of Shigella. This table you don't have to remember it. For the disease, the cause almost always limited to the gastrointestinal tract. So Shigella infection usually is limited to the digestive tract. Shigella are highly communicable. The infective dose is on the order of 10 to the third bacteria. So that means thousands of bacteria can cause disease. That is relatively low compared to like Salmonella. That's low because for Salmonella it needs like 10 to the 9th, 10 to the 8th bacteria to cause infection. But thousands of bacteria is not that low. I used to work on a pathogen called Francisella. The virulent strains of Francisella, 1 to 10 bacteria particle can cause infection and lead to lethal or mortality. So that is even dangerous than this one. That's Francisella, which was developed into bioweapons during the Cold War. One single bacterium can kill a person. That's very powerful. Shigella can invade to the mucosal epithelial cells. They can produce endotoxin, and that's not surprising because they are gram-negative bacteria. They can also produce exotoxin, so that's a Shiga toxin. Then later we'll learn how Shiga toxin works, how it causes disease. This is invasion. Shigella can, from the lumen intestinal space, can infect the M cell. It's a specialized cell. And there, from the M cell, it can further take up by macrophages. And then they lyse the macrophage and infect the epithelial cells again. And when they infect the epithelial cell, they can hijack the machinery of F-actin. So that's the filament actin. The actin filament can drive or push the bacteria to swim inside the cell, or it can push the bacteria to invade the neighboring cell. And this will help the bacteria to spread in the mucosal membrane. So this is the invasion and spreading of the bacteria. And they produce an endotoxin, that's the polysaccharide, and can cause irritation of the bowel wall. Exotoxin, that's the Shiga toxin. Shiga toxin produce diarrhea similar to that of EHEC. It can also inhibit sugar and amino acid absorption. Okay, so that's the enterotoxin. It can also produce the Shiga toxin can also act as a neurotoxin, which can cause other symptoms. For clinical findings, Shiga infection incubation time is relatively short. That is, incubation period is from 1 to 2 days. And within the incubation time, there is no obvious symptoms, so it's not observable. Then the disease will come. The symptoms will show out, like abdominal pain. So that's pain in the stomach, fever, watery diarrhea. Then after that, a day or so later, the number of stools increases with less liquid, but often contains mucus and blood. So, and you can see, if you collect specimen, you can collect the stools and see whether there is mucus and blood. The bowel movement is accompanied by straining and tenesmum. Sorry, I don't know this word. I forgot the meaning of this word. Maybe you can find the information in our textbook. I'll check it later. Okay, this is the clinical findings of Shigella infection. Okay. For diagnosis of Shigella infection, we can check the specimen, like either fresh stool or in the stool you can find mucus flecks, rectal swabs, or in the specimen, if you check it with a microscope, you can find large numbers of fecal leukocytes and some red blood cells. Okay, and that is the characteristic of Shigella infection. Sorry. To culture the bacteria, we can use differential media like McConkey agar or EMB agar, or you can use another agar called selective medium. This one is called Salmonella Shigella agar, and this agar is designed specifically for Salmonella and Shigella, which can select these two genera out. Or you can also use other medium, such as the triple sugar iron agar. I actually never used this medium before. But if you work in a hospital in future, and you will notice that there are so many types of different media or agar you can use. And Shigella infection, you can treat your patient with antibiotics such as ciprofloxacin or ampicillin or other antibiotics. And those antibiotics are commonly inhibitory for Shiga isolates. Okay, so there are some choices. There are many cases that even a person is infected by Shigella, they can recover by themselves. So the disease sometimes is self-limited. So the patient can recover by themselves. Okay. Next one is Salmonella. Salmonella is another famous pathogen. Well, famous in a bad way, because it can cause infection not only in human but in other animals too. General features of Salmonella: they are motile with peritrichous flagella. So you can see here there are so many flagella. They don't ferment lactose or sucrose. When they ferment glucose and mannose, they can form acid and gas. They usually produce this hydrogen sulfur. Hydrogen sulfur, it's a type of gas. And hydrogen sulfur smells like bad eggs. It's very smelly and not pleasant smell. Salmonella can survive in a frozen state, in the frozen water, for a long time. So the sample in the frozen state can let the bacteria to survive for a long time. Salmonella are resistant to certain chemicals, such as brilliant green, sodium deoxycholate, and because these bacteria are resistant to these chemicals, we can actually add those chemicals to the culture media. And if you add these chemicals, they can inhibit growth of other bacteria, and that will let your Salmonella to stand out. So you can use this chemical to make the selective media to culture the mix of bacteria. For antigenic structures of Salmonella, and here's a model of Salmonella cell. It has flagella, so it has H antigen. It's a gram-negative bacteria, so it has lipopolysaccharide. Sorry, this is lipopolysaccharide. It's lipopolysaccharide. It's a wrong spelling. Lipopolysaccharide, it has O antigen. It has capsules. On the capsule, there are virulence factors, so it also has a VI antigen. Here, it's not called K antigen, but VI antigen. It's short for virulence. So these are the different types of antigens. For pathogenesis, there are a lot of Salmonella species which can infect human, such as Salmonella typhi, which is the most important one, and also some Salmonella paratyphi, like paratyphi A and B, are primarily a human pathogen. The majority of Salmonella are chiefly pathogenic in animals. For example, some can only some of them can infect like poultry, pigs, or rodents, cattle, those animals. And because we humans usually consume those products from those animals, such as chicken, pigs, and also cattle. And if the Salmonella can infect those animals, they can contaminate the meat. So that causes the food poisoning in human. They almost always enter the human via oral route. So we contaminate, we eat or drink the contaminated food or drink. That causes entry of the bacteria. They can produce many types of disease in human, including three main types, like enteric fever. So that's infection in the digestive tract, but it increases your body temperature. and also bacteremia with focal lesions. Enterocolitis, that's 3 major types of disease led by Salmonella. And for enterocolitis, that's the most common disease caused by Salmonella infection. Symptoms include nausea, headache, vomiting, profuse diarrhea with a few leukocytes in the stool, and also low-grade level of fever during the enterocolitis. For diagnosis of Salmonella, we need to collect specimens from like blood, stool, urine, and it depends on the progress of the disease. If it's the first week of infection, you can find the bacteria in blood. The second week, you can find it in urine. The second and third week, you should check the stool. And for culture, we can use differential medium like EMB medium, which is also used for Shigella, McConkey medium, or the Salmonella Shigella selective medium. So you can use those different medium for culture of Salmonella. And for a serological test, that means you test the patient's serum, and if the patient is infected by Salmonella, there might be antibody produced, and you can detect the antibody in the patient's serum, and this test is called Widow test. What you do is that you make a serodilution of your patient's serum, and then mix each dilution with Salmonella bacteria. And if there is antibody in the serum, it will react with the antigen on Salmonella and can cause aggregation and can cause agglutination. And this agglutination is obvious with less dilution, like 20-fold dilution, 40-fold dilution. It's obvious. But with the higher dilution, the agglutination it will get lower and lower. And if the dilution is over 100 or 200 times, it's still positive. You can still see the agglutination. We'll see that the test is positive. If there is no agglutination, we'll say that it's negative. And through this, you can determine whether there are antibodies or not in your patient's serum. For treatment, again, you can use different types of antibiotics, including ampicillin and other antibiotics. Okay, so generally we covered the topic of Enterobacteriaceae, and we do have some video covering this material, and I think you can watch by yourself on our Superstar platform. And that's all for today, and thank you. Keep in mind that we'll have a test in several hours. If you want, just take the test as early as possible. Don't wait till the last minute, because you don't know whether you will have some technical problem or not. I will be out of town from tomorrow. I won't be on campus, so probably I cannot help you if you have trouble to take the test. Because we had tests before, probably you will be okay. Okay, so that's all for today. Thank you.